Good afternoon, everyone. I'm Xinting, Field Application Scientist from GenScript European team. GenScript is the world-leading biotech company providing life science services and products. With gene synthesis, peptide, protein, antibody, and preclinical drug development services capabilities, it's my pleasure to introduce our speaker, Dr. Ye. Dr. Ye is the Senior Scientist of R&D Department at GenScript. Today, she wants to talk about precise and efficient non-viral CRISPR gene editing solutions for T-cell engineering. This webinar will take around 40 minutes, and it would be a record section. You can type in your questions in the chat box, and Dr. Ye will answer them after the presentation. If you didn't get your answers during the Q&A section, we will take them down and email back to you. So without further ado, let's welcome Dr. Ye to start the presentation. Dr. Ye, over to you right now.
Hi, Xinting. Thank you very much for the nice introduction. Let me just switch and share my screen. So can you see my screen now?
Yes.
Great. Hello, everyone. Thank you so much for joining us and come to this webinar. In this webinar, the title is Precise and Efficient Non-Viral CRISPR Gene Editing Solutions for T-Cell Engineering. And my name is Dr. Luming Ye. I'm a Senior Scientist from GenScript R&D Department. So in this talk, we'll talk about three topics. The first of all, I will have a brief introduction on T-cell therapy and the CRISPR technology. And then I will go to more detail on non-viral CRISPR gene editing solutions provided by GenScript. Last but not least, I will also give a few tips on transgenic mouse model editing by single-strand DNA templates, because methods and solutions are similar, and also mouse model editing is actually important for drug development and validation. So first of all, there's no doubt that cell therapy is a promising treatment for cancer and infectious disease treatments. So far, there are already 5 CAR T therapies approved by FDA, especially in 2021. In the first few months, there are already 2 CAR T therapies approved by FDA, all from BMS. One is Breyanzi, approved by, I think, early February, which is used to treat large B-cell lymphoma. And at the end of March, there's also another treatment called Abecma, also from Bristol BMS, is designed to treat multiple myeloma. However, all of these 5 approved CAR T therapies are all manufactured by lentiviral virus. The delivery of the CAR gene is done by viral vectors. Actually, with the evolution of gene editing, this method already developed by almost 30 years. It all started at the very beginning that scientists found when there is a double-strand DNA break at the target sites, and the target gene insertion by HDR can be achieved with several multitudes of fold increase. Then researchers started looking for enzymes for gene editing. It started with meganuclease. However, the shift of target sequence can be difficult. Then we start to have artificially engineered nuclease like zinc finger nuclease and TALEN. However, every time when we change the target sequence, to engineer the new enzyme also can be quite cost a lot of time and efforts. It's really until the discovery of CRISPR-Cas9 system. Every time the nuclease is the same, and when we just need to change the new target, we just need to design and synthesize a new guide RNA, and then combine them together, and then we can target any sequence that we want. It's really the convenience and the economic method of this CRISPR-Cas system really revolutionized the way how we editing genes. And also, with the 30 years of development of gene editing, we also realized the tremendous value of CRISPR-Cas system for gene editing. To have a gene knockout, we can just use CRISPR-Cas9 enzyme and the guide RNA to form RNP and cause this double-strand DNA break. And then that double-strand DNA break will be repaired by non-homologic joining mechanism. We also will cause indels and a frame sheet to cause a knockout of a gene. If we also deliver the repair templates with homologic arms, then the double-strand DNA break can be repaired by homology-directed repairing, and therefore we can also insert target gene at the target sequence. Actually, with the fast development of CRISPR-Cas9 methods, there are already several of these therapeutic methods being reached to clinical phase. This table actually take from a review paper from published at early 2020. In this table, there are 12 therapies listed. One of them is made by meganuclease methods, and 5 of them are actually engineered by Thailand, and 6 already is the therapy were developed via CRISPR-Cas9 system. Especially for this one, this NYC T-cell therapy. Its phase 1 clinical trial data already published in early 2020 and proved that CRISPR engineered T cells are safe for disease treatment. However, when we look at this, how this cell therapy was engineered, CRISPR-Cas9 system was only used to do the gene knockdown to remove the endogenous TCR and also PD-1 gene. The delivery of the TCR gene is still delivered by lentiviral vector. We definitely can combine CRISPR-Cas system and the viral vectors to engineer T-cell therapies. Like first use the viral vector to deliver the CAR or TCR gene, and then use CRISPR system to remove the other targets. However, with CRISPR-Cas system, we really can remove the TCR or other gene figures and also insert or transfer the CAR sequence by one step. Also with this method, more and more type of cell therapies are developed, far more beyond CAR T therapies. There are also CAR macrophage or CAR NK cells, and also CAR B cells. All benefits from this convenient and easy cell engineering methods. Also, compared to viral transduction methods, non-viral CRISPR gene editing methods have also several advantages. First of all, compared to viral transduction, the cell engineering process is much easier and simplified. The other huge advantage is the precision. The knocking of CAR or TCR gene can be precisely engineered at the target sequence by 2 steps. One is secured by the Cas9 and RNP system to do the precise double-strand DNA break. And the other one is the gene knock-in by homology-directed repairing. Also with the precise gene editing, now we can insert the CAR sequence or TCR sequence right at the beginning of track gene exon and the very beginning of the track gene. Therefore, we can drive the expression of CAR or TCR, but in the endogenous promoter sequence. Not like viral methods, we have to drive the CAR expression by a external promoter, which may cause overexpression or like the cell exhaustion. Also, for editing efficiency, I think at the end of last year, still viral transduction can make about 60% of CAR expression efficiency. However, non-viral CRISPR editing may be still a little bit low, only about 20%. However, with several groups' tremendous work, now we also see reports that by non-viral methods, the CAR gene insertion at the track locus can achieve like 40 to 50, even 50%. Also compares to viral transduction methods, the non-viral CRISPR gene editing also reduce, and even minimize off-target risk, especially when you carefully select the right repairing templates like single-strand DNA. We can even further minimize the off-target risk. Last but not the end is also the manufacture process, both for the key materials like Cas9 nuclease, single-guide RNA, and the DNA repair templates. Their manufacture process are all much easier compared to viral vector preparation. Also, when you use non-viral methods to engineer the T cell, because there's no viral material related, the entire GMP facility and the manufacture process can be much easier. This is the general workflow for CRISPR-based non-viral gene editing process. First, we need to have design a synthesized guide RNA and combine it with Cas9 nuclease to form RNP. And then we also add the HDR templates, which is usually is double-strand DNA or single-strand DNA. And together, we mix with isolated and activated T cell and do electroporation. After cell expansion and recover, we analyze the final results with FACS to see the knock-in efficiency. Now I will take a few more minutes to discuss about these key materials in detail. The first key material is actually the Cas9 nuclease. To choose Cas9 nuclease with nuclear location signal can improve the nuclear transfection efficiency dramatically. Also, at the early stage of your research, it will be great to choose the Cas9 nuclease with EGFP tag. In this way, we can monitor the transfection efficiency of the RNP complex. In this figure, you can see that when we use 7.5 picomolar, 10 picomolar, even up to 20 picomolar of the RNP complex, the nuclear transfection efficiency increases linearly. Also, right now, we are also very glad that we have licensed the enhanced version of Cas9 protein with nuclear location signal modified. For this version of the Cas9 nuclease, we found that compared to the wild-type Cas9, the knockout efficiency is increased. In this experiment, we use Cas9 RNP to do the track gene knockout. Therefore, the higher of the enzyme activity, the lower of the track gene expression. Especially when we add the double-strand DNA HDR templates is added in this electroporation system. It's also often we found that with double-strand DNA or single-strand DNA repair templates added, the RNP activity will be compromised. As you can see here, in the wild-type group, the track expression is increased. However, in this the enhanced version of Cas9 group, the knockout efficiency maintains all at a very high level. Also, with not only the high knockout efficiency, we also found that this enhanced version of Cas9 can also increase the knock-in efficiency. This is when we compare to add 40 picomolar wild-type Cas9 and 40 picomolar of enhanced Cas9, and we try to insert a GFP tag at RAB11A in the RAB11A gene. We found with enhanced version of Cas9, the knock-in efficiency or GFP positive rate reached to 35%, but the wild type only gets 26%. It might due to the high knockout efficiency of EISP Cas9. The second key material is the guide RNA. Here I collect 3 different articles. They all report the result of the PD-1 gene knockout by CRISPR-Cas system, but different version of guide RNA formats. On the left is actually a clinical result published last year. And there they engineered T cell with CRISPR-Cas9 system to knock out PD-1 gene. However, the authors, because this work was done by a few years ago, at that time, there is no single-guide RNA or purified Cas9 nuclease. So the researchers chose to use plasmid to co-encode the Cas9 protein and guide RNA and deliver into the T cell and let them to transcribe in the cell. In this work, the researchers published that the mean PD-1 gene knockout efficiency is about only 10%. In the middle is the article that I mentioned before that proved the CRISPR engineered T cell is safe. In that article, the author also tried to knock out a few genes like the TRAC and TRBC and also PD-1 gene. In this work, the author used the in vitro transcribed single-guide RNA. And the PD-1 gene knockout efficiency is about 20%. On the right is actually an article on medRxiv by the end of last year. It's done by a cell therapy group in China, Shanghai. The research group, the authors also try to knock in the anti-CD19 CAR into the PD-1 gene locus. And with the Cas9 protein and the safe edit sgRNA from GenScript, the authors measured the indel percentage of PD-1 genes about 80%. This is shown by the ELISA antibody presentation. Also, the PD-1 gene expression is only about a few percentage, which means knockout efficiency is more than 80%. At GenScript, we offer easy edit and further purified safe edit single-guide RNA. They all have default modifications with 3 oxy, maxi, and a phosphothioate modification for better stability. And we have very flex sgRNA design to work with different kinds of Cas9 proteins. Also, for safe edit sgRNA, we have further HPLC purified to ensure more than 90% purity. On this figure, that we can see that the safe edit sgRNA has more than 99% of purity and has much more better clearance compared to the sgRNA purchased from all competitors. And on the right figure, that we test sgRNA at track locus, and we test different dosages like 7.5, 10, and 20 picomole, two different type of proteins, and the knockout efficiency all above like 90%, and also all maintain very high cell viability. For the DNA payloads for HDR templates, at the very beginning, we also wonder which is the best template format. So we designed a knock-in templates flanking the track locus, and also because we don't have the antigen to detect NY-ESO TCR, so we just swap that TCR gene with a CMV promoter drive EGFP gene. With this template, we also make it into different formats like plasmid DNA and mini-circle DNA. Both of them are circular double-strand DNA templates. Also, we made PCR double-strand DNA, which is linear double-strand DNA templates, and also single-strand DNA. We transfer this different type of DNA payloads together with RMP into heterogeneous T cell line. 2 days after electroporation, we found that almost all of the different of DNA payloads have quite high GFP positive rates, like 40%. However, for the non-RMP group, control group, this is the group that we only put the plasmid or other type of DNA payloads, but no RMP, which means no double-strand DNA break. In this control break group, we also found a very high GFP expression level, which can be the background. So we measure the experiment groups 7 days later again after electroporation. In this moment, we found that for plasmid DNA and mini-circle, this type of circular DNA templates, there's barely any difference between the testing group and the control group, which indicates there might be a very limited amount of knock-in events. For PCR double-strand DNA, we actually found that there's still a certain level of GFP positive rate background, but still there's quite a significant difference between the sample testing group and the control group. For single-strand DNA, at the same amount, like 2 micrograms, the knock-in efficiency or GFP expression efficiency is not very high. However, in the non-RMP group, the background is very low, even at day 2. So we found that linear DNA payloads have better performance compared to circular DNA templates. And at the same dosage level, like 2 micrograms, single-strand DNA had lower knock-in efficiency, but also low false positive signals. Therefore, we decided to continue with this single-strand DNA and double-strand DNA, these 2 types of linear templates, to continue develop them for non-viral CRISPR gene editing. We developed precision single-strand DNA for a precise knock-in for cell therapy, and also for transgenic animal model generation, because it's every time you see the positive signal, it's accurate editing. There's no NHEJ risk or other false positive signals. Also, we developed stable double-strand DNA templates for knock-in templates for ex vivo cell therapy, and also can be used for expression vehicle for cell and gene therapy. In this figure, we did some in-house testing to compare the single-strand DNA with double-strand DNA. And this time, we can even test a higher dosage. Still, in this test, we insert the GFP at RAB11A locus, and we tested on a primary T cell isolated from ALL patients, and by Lonza 2B nuclear factor. In this case, we found that compared to PCR purified double-strand DNA, single-strand DNA has much higher survivability, which means that it has much lower toxicity. Like at 4 microgram, no RMP control group, in day 6, the survivability is only about 27%. However, single-strand DNA is doubled about the survivability. And also, when we add RMP in the testing group for electroporation, in double-strand DNA group, the survivability decreases linearly with the increase of DNA payload dosage. For single-strand DNA, the survivability maintains at a very high level, even at 4 micrograms. And the knock-in efficiency is even more interesting. At low amount, like 1 microgram or 2 micrograms, double-strand DNA templates gives slightly higher GFP positive rate compared to single-strand DNA. However, at the 4 micrograms group, we found there's no GFP positive rate for double-strand DNA samples because the cell dies dramatically and the status of the cell is not good anymore. However, for single-strand DNA at 4 micrograms, we see the GFP positive rate increases from 10% to 26%, increase 2.6 times when we just increase the templates to 2 times, which means single-strand DNA can be a good template when the cell is sensitive to double-strand DNA, and we can achieve high survivability cell engineering efficiency by increasing the template dosage. Also, by an article published still on bioRxiv in 2019, the authors use a long-read sequencing, they found that when compares to when we use PCR purified amplicon, which has free 3-pair end hydroxy group, this type of blonde and double-strand DNA templates, the perfect HDR rate is relatively low, and there are also different kind of truncations and non-perfect engineering events present in the engineered cell, like duplication of homology arm, which indicates the double-strand DNA is actually ligated to the double-strand DNA break by NHEJ. And also, there also have insertions of concateners, which means the double-strand DNA ligated to a cell first from a concatener and then be engineered into the double-strand DNA break. While with single-strand DNA templates, the percentage of the perfect HDR rate is very high. However, there's still a few cases, a low risk of truncated insertion, because when single-strand DNA is not modified, it can be digested by the isonuclease in the cell, which may cause truncated insertion. And also, we also developed the stable double-strand DNA. We still use this CME promoter drug EGFP templates, and we compare the stable double-strand DNA with also with purified PCR amplicon. Still, this time we test a T cell from a healthy donor. For this cell sample, when we use the double-strand DNA 2 micrograms, the editing efficiency is only about 2.6%. When we use stable dsDNA, even at day 5 observation, we found at 1 microgram we can reach 8.3% of knock-in efficiency, and at 2 microgram dosage we can achieve 11% efficiency. However, for double-strand DNA, it's also known that it has immune genicity to T cell and is toxic to the cell. So at 4 microgram, still there's no JAK positive events anymore because the cell status is not good anymore. To summarize these two kind of materials, we have precision single-strand DNA, and it's produced by our proprietary isothermal amplification methods. It started from sequence verified templates, and it's produced by our in-house produced high-fidelity DNA polymerase. It's very easy to scale up because our production method is based on isothermal amplification methods. And we are the experts in difficult sequences. It's available from the length range is 150 nucleotide long to 5,000 nucleotide long. And for like sequence around 3 kb, we can produce around or less than 3 kb. We can produce 2 milligrams per batch. For stable dsDNA, it's also produced by our proprietary vector and production process. And it's actually produced from the plasmid, and we have the capability to produce large scale of plasmids. And then we'll harvest the target gene by in-house produced animal-free enzymes. And also the entire reaction system is animal-free. The stable dsDNA is available from 1.5 kb to 10 kb, up to 3 milligrams per batch. This slide is a few examples of a single-strand DNA that we delivered to previous orders. From a few hundred nucleotide long to almost 4 kb, we can all produce them in good quality. And no matter the high GC content or high repetitive sequence, we are all able to deal with them. And also for each of the single-strand DNA, there's no other bands present in the final product. And there's also no double-strand DNA residues left in this final product. In the end of 2019, there are actually 2 articles published back-to-back. They all prove that by modified double-strand DNA at 5-pair end, they can have higher knock-in efficiency, because by modifying the 5-pair end and 3-pair end double-strand DNA, the DNA can be protected from the isonuclease in the cell and enhance the stability. And then the knock-in efficiency can be increased. By this knowledge, And also, we know that the drawback of blunt and linear DNA like PCR amplicon or linearized plasmid DNA. We developed a stable linear DNA. It contains the gene of interest, and at both ends there are 28 BP extra sequence, and at the end of each end, the double-strand DNA is closed covalently, so can make this DNA very stable and prevent NHEJ. The advantage of this double-strand DNA product is its starting material is maxi-prepped plasmid. It's the best solution to ensure sequence integrity, and is suitable for cell banking or production vector banking to ensure the lot consistency and can be transferred to compliant manufacturer. And the entire production process is animal-free, mammalian cell-free, and BS-free. And we also prove this stable linear double-strand DNA. It actually has much less, or basically zero, plasmid residue compared to PCR amplicon or linearized plasmid. And also, in the end, the covalently closed double-strand DNA ends can prevent non-homologous end joining risk. Here are a few examples of a double-strand DNA product that we delivered before, like this example of a 2.6 kb sequence, 3.8 kb, 5.6 kb, and 6.0 kb. They all produce in very high quantity and good quality. And also for this, both single-strand DNA, double-strand DNA materials, now we have preclinical grade DNA payloads materials available, also with extra QC methods to ensure safety. Besides the measurement, QC measurements of this product, like appearance, quantity, and purity, and sequence integrity, we also can add on other QC methods to ensure the biosafety, like bioburden, stability test, to test the protein residue in the toxin level, DNA's residue, RNA's contamination. We can also measure the pH and conductivity of the aqueous solution, and we can also offer stability tests upon request. Besides the three key materials, there are actually a lot of other key elements that you need to test and ensure at bench to achieve both high knock-in efficiency and high cell viability. This includes donor cell selection. We found some of the cells are very vulnerable to DNA material or electroporation treatment, but some of them are quite robust. You often get nice results with these robust cell samples. And also, you need to fine-tune the electroporation program, especially for CRISPR gene editing. We need to deliver the Cas9 and the sgRNA RNP complex and donor templates into the nucleus, which is much more difficult and complicated than we transfer a plasmid into the cell for transit expression. And also, we can choose the knock-in template, either by double-strand DNA or single-strand DNA, or with any type of modifications. Also, the length of homology arm is also, we can have some tests to ensure the high knock-in efficiency. But normally, we can choose from 300 to 500 nucleotide long. Also, we can fine-tune the Cas9 and guide RNA ratio to ensure the highest knockout efficiency. Usually, it can be 1 versus 1, or even to 1 versus 3. But the precise ratio can be different at different targets. And also, we can choose different type of targets to have higher knock-in efficiency, or use chemical HDR enhancers to have higher knock-in efficiency, especially when you're working on cell line engineering. And also, we can use some polymers like PGA to be used as electroporation enhancer to achieve higher survivability and help the cell against the electroporation reaction. In summary, to achieve high knock-in efficiency and survivability is quite complicated, and let GenScript to take care of the other reagents solutions. In the end, with all of these developing methods and then keep on published results, CRISPR system methods have already been used in several different fields. We just talked about cell therapy, and in the next few minutes, I will also talk about how it's being used for animal model generation. This is a case that one of our collaborators on GenPharmaTech at the very beginning of last year, when we just had the outbreak of COVID-19 in China, GenPharmaTech ordered a 3.6 kb single-strand DNA to use CRISPR method to knock in the humanized ACE2 CDS at the mouse ACE2 exon. And with the single-strand DNA, GenPharmaTech was able to achieve the accurate engineered mouse by one round of experiments. And this humanized ACE2 mouse was available since just a few months, like in June 2020, which can contribute to the study of COVID-19 disease. Actually, when we use CRISPR method to engineer a transgenic mouse model, there are also a few issues. Some of the issue we heard a lot from our clients is point mutation issue. It's actually happened in the same in this humanized ACE2 mouse generation as well. Our clients or collaborator feedback that after the engineer microinjection, they acquired 17 puppies. 12 of them had full-length integration detected by PCR methods. And after sequencing, they found only 5 of them are accurately modified as they designed. The rest of them all have point mutations at different sites and different types. They wonder, is this caused by the quality of single-strand DNA? However, single-strand DNA is actually quantified by sequencing the single-strand DNA itself with a reverse primer, and we're sure there's no hint of cross-peak or mutation risk with the sequencing file. And our clients agree the same. And we think the point mutations were not caused by the quality of single-strand DNA, but the single-strand DNA specific cytidine deaminase in the cell, which can induce mutations at the single-strand DNA and around the double-strand DNA break region. And this is a article published in 2017, shows that the APOBAC, which was famous for base editor, it actually can cause base substitution mutation on single-strand DNA, single SSODN. The authors found that in this APOBAC gene overexpressed cell line, the mutation rate increases dramatically compared to the normal hypotonic FT cell line. And also, especially when we found most of the point mutations actually happens at the homology arm region, and this mutation cannot be caused by the HDR repair templates, because the templates only provide the knock-in region contents. When we found these mutations at the homology arm region, it's actually because the APOBAC can mediate base substitution mutations, not only in SSODN, but also at the double-strand DNA break region, when there's also single-strand DNA present at double-strand DNA break. The authors suggest to use double-strand DNA as a repair template. However, we already know that double-strand DNA can cause other issues like duplication of a homology arm or insertion of concatenamers. So it's still better to use single-strand DNA. But there's also another issue when we use single-strand DNA as repair templates. That is, quite often we also heard that there will be file pair and truncation found due to the single-strand DNA can be digested in the cell by isonuclease. Because when there's double-strand DNA strand break, the single-strand DNA repair template will anneal at the other side, and the 3' end of single-strand DNA will anneal first, then it be protected. And it's only when we repair along these single-strand DNA templates when there's missing region at file parent, it will cause parent truncation. To solve this problem, we will work together with Professor Masato Otsuka from Tokai University. We tested normal single-strand DNA and also five-pair and modified single-strand DNA to see whether the modified single-strand DNA can prevent this truncation and mutations. Professor Masato Otsuka tests two methods. One is Abecma, the other is microinjection. And with both DNA delivery method, we found that the modified single-strand DNA can have a much higher number of obtained copies. And also, when we have this sequence verified, the modified single-strand DNA can provide a much higher positive, complete accurate engineered mouse compared to normal single-strand DNA. Also, by microinjection methods, the completely accurate engineered mouse is 8 to 9 copies, while normal single-strand DNA can only achieve 2 to 6 complete accurate engineered mouse. And that's all for today's talk. Thank you so much for listening. If you have any questions or requests on the special designed materials, please feel free to ask. And we are really happy to have able to support all the supports. Thank you very much.
Okay, thanks, Dr. Ye, for the great presentation. So we will move to Q&A section right now. The first question, compared with IVT sgRNA, what are the advantages of chemical synthesis sgRNA?
Okay, actually, that's a very good question. I think first of all, there are two major advantages, three actually. One is for chemically synthesized sgRNA, you can actually just order from any type of oligosynthesis vendors, and you can have the single sgRNA sent to you directly can be used. And the other thing is like chemical synthesized sgRNA can add 5' end and 3' end modification to increase the stability. Well, we can't add these type of modifications with IVT methods. And also, actually there's a disadvantage of IVT method produced sgRNA because it's produced by transcriptase. At the 5' end of the sgRNA, there'll be a triphosphate group. It actually proved has immunogenicity problem, which can actually trigger the immune response in the cell. So it's not, that's one issue. And the other issue is like when we use IVT method to produce sgRNA, quite often there'll be extra G at the 5' end. And actually, there was an NAR paper in last year. It's proved that this extra G at 5' end can cause off-target or mismatch risks. So compare in all of this conclusion, the chemically synthesized sgRNA has much more advantage than IVT produced sgRNA.
Okay, thanks, Dr. Ye. So the next question, could you please elaborate a bit on how to improve the efficiency of gene editing with non-viral CRISPR system from your experience?
Oh, wow, that's actually a very big question. I think for my experience, I think it's really important to start with a nice cell donor sample, especially when you are working with, for example, allogenic cell therapy development. Then you have quite some time and resource to test different donors of T cell and find the most easy one to be manipulated. Actually, if you take a look at when you read the article published by Theodore Ruth from Marston's lab, like the Nature paper in 2018, you can find out there are actually always two data sets of T cell engineered results. One of them is often give much higher knocking efficiency and also higher cell viability, while the other one is much lower editing efficiency, also the same lower cell efficiency. They actually what we found that it's just because the cell itself are a different kind of tolerance to this CRISPR engineering and the electroporation treatments. So it's really important to start with a good cell sample. And the other thing is that they actually need a lot of fine-tuning steps to make sure the Cas9 and RNP sgRNA ratio and DNA templates and find the best electroporation program. It can't be too harsh. It will reduce the cell viability. It also cannot be too mild because it won't deliver the RNP and the donor into the nucleus. And then I think other than that, I think it will be, we can reach to like 20% of knocking efficiency, especially with the several already quite published locus like RAB11A or track locus. Or you can, I also think it's a nice idea to test the published protocols and published locus first to fine tune your system. And then you can, when everything's set and can reach the published data level, then you can start to test your own design. And if you need some testing samples, control samples, we're also happy to provide.
Okay, thanks, Dr. Ye. So the next question, do you have any suggestions on how to qualification of genome editing efficiency?
Sorry, how to
qualification of genome editing efficiency?
Okay. Yes, actually, for for example, for cell engineering, I think the best way is to quantify it by flow cytometry. For example, like we show that for when we want to knock out the track gene, then we can use the this TCR antibody to show the knockout efficiency. Also, it's also actually quite easier to monitor or quantify the knockout efficiency. One is by the other way is to just amplify the knockout region, just a few left and right, like a few 100 nucleotide long, and amplify it, and then send it to Sanger sequencing, and then analyze the amplicom by, there are several softwares. Then we can quantify the indel ratio and to kind of guess the knockout efficiency. Actually, this method is pretty accurate to NGS. Can be quite aligned to NGS method in our house test. So it's quite reliable. And of course, you really want to know the knockout efficiency. The best way is to use antibody to present whether the gene is really knocked out and is not expressed, and check it on flow cytometry. And for knocking efficiency, I think for if you want just use a small SSODN to introduce a point mutation or a LuxP tag, you can definitely quantify by PCR methods. However, if you knock a CAR sequence, it's really long, and the knock efficiency can be low at some time. Still, the best way is actually to present this the CAR that you designed by the special antigen and special with tag and quantify on FACS.
Okay, thanks, Dr. Ye. So the next question, except the T cell, are there other types of cells can be edited efficiently by non-viral CRISPR technique?
Yes, actually, there are actually quite a few other type of cell therapies are engineered by non-viral cell engineering type methods. For example, stem cells, iPSC stem cells, and also there are also a few studies on B cell and NK cell, and they all started with using non-viral CRISPR gene editing methods.
Okay, thanks, Dr. Ye. So the next question, is it true that GenScript safe-edit sgRNA is safety and precision method for genome editing?
Yes, that's a definite yes. We actually already tested quite a few locus to prove it's accurate, and because also the high purity can prevent this truncated SGI to be targeted at different locus. So we actually also did validation by NGS-based IGAT-seq methods to show that with carefully designed sgRNA sequence, we wouldn't detect off-target events.
Okay, thanks, Dr. Ye. So the next question, if we can produce knock-ins without homologous arms?
You can. I think there are also other methods called microhomology arm and engineering methods. Some of the reports say that it has much higher efficiency, but I think if you do the knock-in templates, design templates completely without homology arm, then it'll be a very high risk to this templates will be inserted to other place DNA double-strand DNA break, and this only works with blunt ends dsDNA templates, which I wouldn't recommend.
Okay, thanks, Dr. Ye. Thanks for your great presentation, and also to all who joined the webinar today. Due to the time constraint, we just end the Q&A section here. For the questions that we didn't have time to answer, I will email back all the answers to you from Dr. Ye to you. And I hope you find the section useful and beneficial for your future work. Additionally, we have prepared another 2 free webinars in May about introducing CRISPR technologies and the CHO expressing platform. Please follow us in LinkedIn to stay tuned for the latest webinar schedule. And thank you to everyone for joining again, and have a nice day. Bye-bye.
Bye-bye.
Bye-bye.